lecture series, Science Times, Exploring the Frontiers of Science. We're delighted to see you all here tonight, and you may be interested to know that President Shirley Tillman helped us put this lecture series together, and she will, as I'm sure you know, she will be speaking in a few weeks. So we worked with her, and she helped us pick out an absolute wonderful lineup of speakers and topics. We want to tell you a little bit about Erin Lemenick before Professor Murphy starts her talk. Erin Lemenick was a Princeton University physics professor, and this series is named in his honor. He was beloved by generations of students here at the university. He was known as a distinguished teacher and scholar and was deeply devoted to science education. He was also instrumental in founding Princeton's Women's Studies program and also its Department of Molecular Biology. Of course, we at Princeton Adult School remember him most fondly because he served on our board for many years and was responsible for creating many well-loved lecture series and courses on science. We've asked Erin's son, Michael Lemenick, to say a few words about his dad, and I'm going to say a few words about Michael, and then he'll come up and talk about Erin. Michael Lemenick is the senior staff writer at Climate Central and a former senior science writer for Time magazine. He's also written for Discover magazine, Yale Environment 360, Scientific American, and others, and has written a number of popular books on science and astrophysics. Just as his father did, Michael has helped the adult school create courses on science and deliver them. He teaches communications and journalism at Princeton University, and he makes his home here in Princeton. So please welcome Erin's son, Michael.
Okay, so yes, that's how many people think of me, Erin's son, and I don't look something like him, so we've got the complete package here, and sound like him too. So I will just say a couple of things very, very quickly. First of all, I am honored to be able to introduce this series. My father would be deeply honored to know that it had been named for him. He would be, in fact, if he were here, he'd be sitting in the front row sobbing because he could get very emotional about things like this. So I'll just say that it's really, really appropriate that the series has been named in his honor because there were three things he deeply loved. One was science, deeply loved science. He was so much in love with science that he could not resist teaching about it. He would grab you by the lapels, essentially, and tell you about science. And finally, he loved Princeton very much. As for the teaching, I first experienced it probably at the age of 4 or 5. We would be driving back from Philadelphia after visiting his parents or my mother's parents, and in the darkness of the car, he would tell stories about science to these kids sitting in the back, and they were so engaging and exciting, and his excitement came through so much that I determined at a very early age that I would become a scientist like him. And that actually didn't work out so well because there was a lot of math involved, and he never mentioned that. But I became a science journalist instead, so I've been able to stay in touch with the world of science and to appreciate it, maybe not as deeply as he did, but I love it very much myself. He was, as you've heard, a very beloved teacher, and that's what they always say at these things. But in fact, people, men with gray hair, come up to me, and it's men because he taught at Haverford College, which was all male in those days, and at Princeton in the early days was also all male. So men with gray hair come up to me and talk about how deeply they loved his courses in the 50s and 60s. This still happens to me. He just could not resist, and when he retired and had to step down from had to retire from the faculty, he couldn't let go of it. So he became involved with the adult school. He became involved with the teacher preparation program at Princeton, the summer program. He could not stop teaching until the very end. So he loved that deeply. And finally, Princeton meant more to him than almost anything. He loved the institution, and he would be especially gratified that this series is populated entirely by Princeton faculty members. So I think that's all you need to hear from me. I want to get to the main event, and I know that you're going to enjoy the series. I won't say that I hope you will, and I look forward to it myself. Thank you.
Thank you so much. Now you have a flavor of the reason why we decided to call this the Aaron Lemonek series. I'd like to say a little bit about Professor Colleen Murphy and then let her take over. She received her BS in Biophysical and Biochemical Sciences from the University of Houston and her PhD in Biochemistry from Stanford University. As an LSRF and Ellison postdoctoral fellow at UC San Francisco, she studied the transcriptional regulation of C. elegans longevity. C. elegans are nematodes, and that's all I know about them. But she knows a lot about them. In her own lab at Princeton, she has focused on early aging phenotypes, including reproductive cessation and age-related decline of learning and memory. No one in this room has that problem, but we can be polite and listen anyway. Developing quantitative assays of these behaviors. Dr. Murphy has received Young Scholar Awards from the Sloan Foundation, the March of Dimes, the Pew Foundation, the Keck Fund, the McKnight Foundation, an NIH New Innovator, and the Glenn Foundation. And she's one of the first names that President Tillman, well, Professor Tillman as well, suggested for our series, and she said yes right away. So I'm very excited, and please welcome Dr. Murphy.
Grateful for the invitation to come and speak to you. You'll gather as I talk, I love talking about my work. Yeah, it's too dark. So anyway, let me start by saying, yes, my lab, we study aging, and we try to use a little bit lighter. I don't want to bore you when they fall asleep in the lab. And I'm going to tell you all about C. elegans, so by the time we're done, you'll actually know something about it, and why we use this as our model system instead of using something like this. All right, so why should we study aging? Not just an interesting philosophical question, but as a population, our average age is rising, and that's a good thing in general. But of course, along with aging comes a lot of age-related diseases, right? And so these are each worth studying individually, but our thought is that if we can slow down the process of aging, we might at least be able to stave off some of these diseases until later in life. All right, so what do we consider old? Well, it kind of depends on what you're doing, right? If you're a professional athlete, your longevity actually is much shorter, right? In your early 40s, that would be considered long-lived. If you're a gymnast, of course, it's even worse. You're washed up by 20. But if you're a scientist, which is why you can last for a long time. So the message of my talk largely is about function, right? It's really being old depends on what function we're talking about. All right, so how can we study aging? And this will explain to you a little bit about why we chose C. elegans, this little nematode I'm going to show you about. All right, so here's a scale. We have something that is the simplest organism and has a short lifespan, all the way up to humans, right? Very complex, long lifespan. And we have this slew of organisms in between, and all of these are experimental models that we could use. From bacteria through yeast, these are single-celled organisms. This is C. elegans, what I'm studying, so these are flies. You'll hear about this from Mala Murthy later in the series. Zebrafish they live a little bit longer, a couple of years. Months, mice, so a lot of people study mice for aging, but for our purposes, this would take about half of a graduate student's time. So I don't really like mice until you actually know what you want to study. And so of course monkeys can live decades. So if you really are sure about what you want to study, you can study that. But we chose C. elegans because it seems like a happy medium. We can do experiments very quickly on the order of days to weeks. But because it's relatively complex and a multicellular organism, we can actually learn a lot that we think we could apply to higher organisms. All right, so what's C. elegans? So these are small nematodes. This is a millimeter long. But we can really see all its tissues nicely. You can see that you can actually see individual organs. They have very simple body, but they have an intestine, a mouth, muscle, and they grow very quickly. So they go from an egg to an adult in about 2.5 days. And they have about 300 progeny types. So you can get a lot of these little guys really quickly. As I said, they're transparent, so you can see all their tissues. they have what's called an invariant lineage, and this is actually kind of remarkable. So each one of these organisms, each one of these animals only has exactly 959 cells. So this has been traced, so the people who worked in worms very early on, they followed each cell division, and that's how they learned about a very important process called programmed cell death. But each one of these has exactly 959 cells, and each cell has been named, and so we can actually follow the fate of that cell very clearly. And so for that reason, and for other reasons, it's a very good genetic and genomic system. Now for the aging field, it's really great because it only lives 2 to 3 weeks. Right? So that means we can do a lot of experiments very quickly, and within that period of time, it undergoes some very obvious aging defects. Okay, so they slow down. Here, these worms on day 4, they're moving very nicely. By this day, they're barely moving. They accumulate actually some fat pigments in their guts, and their muscles start to fall apart, and they get very wrinkly. So they have some very, very familiar feeling phenotypes. And of course, the reason we use worms is because of evolution, we actually can say that the genes that we study in worms, many of them actually have homologs in higher organisms, including ourselves. And so we hope that it's direct relevance. All right, so the real thing that broke open this field was the work that my postdoctoral mentor, Cynthia Kenya, did back in the early 90s. And she found she made this important discovery, and that is, so I'm going to show you a lot of these are called survival curves. They're Kaplan-Meier survival curves. So this basically means a population of animals, we follow them, and they're living, and then as they die, we mark them off, okay? And so wild-type worms shown in black will live, as I said, 2 to 3 weeks, with mean lifespan. These DAF2 mutants, and I'll tell you more about those, live twice as long. And not only do they live twice as long, but they are really youthful looking and healthy during that time. And so that's what Cynthia discovered. And then later, they cloned this gene and found out that it was an insulin receptor. Okay? So in worms, this is the only insulin receptor. It has a good amount of similarity to the human insulin and IGF-1-like receptors. Okay? So that's a really direct feeling. If this had turned out to be something else, we might not have been as interested, but this really felt like something very important. All right, so they already knew there was a gene that acted downstream of it called DAF16. So let me tell you about this. So in genetic terms, what we'd say is the DAF2 normally prevents longevity. So when you have a mutant, that blocks it. That's what this little T means. Okay? And DAF16 acts downstream of it, and it's required for that longevity. So when you knock out that gene, you can see now it's short-lived again. Okay? So that says, in genetic terms, that DAF2 normally blocks DAF16's activity, and DAF16's role is to promote longevity. Okay? So that's the genetic shorthand that we use. And DAF16 turned out to be what's called a transcription factor. That means it's a protein that binds to DNA, and it directs the genes that get turned on. All right? And so just to drive home this point about similarity, so these are some of the genes I've told you about in this pathway, and I'll tell you more. But what you should notice that these same genes occur in other organisms, including mammals. So we have these insulin IGF-1-like peptides that act through this receptor I just told you about, DAF2. Those signal through a kinase pathway that I'll tell you a little bit more about, and they end up at this transcription factor. So our hope is that whatever is downstream of the thing in C. elegans will tell us something about what's going on in other systems. All right, so I told you about DAF2, this insulin receptor, and how it blocks DAF16 activity. And the way it works is through a whole mess of other genes, and basically what these proteins do is they direct the phosphorylation of DAF16. And really what phosphorylation is the mark for what that protein should be doing. And in this case, these little yellow balls is phosphorylation. So when this is happening, then these worms have a normal lifespan, like I showed you, that black line. And they go through normal development. But when you block this through any of these mutants in this pathway, now DAF16 doesn't have that phosphorylation mark, and that allows it to go into the nucleus. Okay? And as I said, it's a transcription factor. So what that means is it can do its job now. So when it goes into the nucleus, it can bind to DNA, and it can turn on gene expression. All right, so under these conditions, that's when we see that long lifespan of the DAF2 worms. And these worms, I'm going to tell you about my lab's findings, where we found that they have an extended reproductive span, and they delay cognitive decline. All right, so one of the things we wanted to know right away was, what are the genes that DAF16 regulates? Because that seems important, right? We've got all these things, but we don't know what's downstream. And so to do that, as a postdoc in Cynthia's lab, what I did was a lot of microarray experiments. So we basically use these DNA spots. And I won't explain this too much, except for me to say that red is up. So these are genes that go up in those long-lived DAF2 mutants, and they go down in a DAF16 mutant. So that's the set of genes I was really interested in. And we tested those using a technique called RNAi, where we can knock down that gene's function individually. And so the only thing to show you here is just that this black line here is DAF2's long lifespan. And when we take away any one of these genes, it shortens it a little bit. Okay? This was important to me because it meant that this blue line is DAF16. So none of these genes act just like DAF16. When you take it away, they don't cause the entire failure of the lifespan. Instead, it seems like DAF16 is directing a whole slew of genes that cumulatively add up to give a long lifespan. So that's why DAF16 is so important, is because it has this huge regulatory role. It's a master regulator. And so the genes that it regulates kind of make sense to us. When we looked at the list, we finally understood why these worms are living long. It's because these worms do a good job of taking care of all the, I told you they have these cells. That's the only cells they ever get. And the way they keep those cells living long is by taking care of the proteins inside those cells. So that is they take care of protein folding. They don't let the proteins get all gummed up. They fight against all types of different stresses. So if there's oxidative stress, they turn on genes that fight against that. All kinds of heavy metals, anything they're exposed to in their environment. This DAF16 turns on a gene that basically stamps that out. And then over here we have a bunch of lifespan shortening genes, and we don't understand those at all. We're still trying to understand how it is that the organism would turn on a gene that normally would start to kill it. But they do that. And then we found also these signaling regulators that basically DAF16 turns on insulin-like peptides that go back and signal to DAF2 and sort of feed forward to tell the other cells to live long. So that was what we knew before this work. So the take-home message from this work is that these longevity-regulated targets kind of answer half the things that we have to do. Like, our body has cells that turn over and cells that don't turn over. And the cells that don't turn over have to do a good job of keeping their proteins healthy, because otherwise they're not going to get another chance. That cell will just die. And that's what these worms do a really great job of. All right, so all the work I told you about up till now is just about looking at how long the animal survives. But we know that those DAF2 worms really are healthy and youthful for a long time. And I'm sorry, I actually have a movie of this, so if you have time afterwards, I can show you a little demonstration of this. But we all know that just basic survival is not the same thing as living well, right? But what you really want to look at is all these different things that change with age and try to track those, right? And then one of the questions we have is, if we could find the earliest decline with age, if we managed to do things, manipulations that would extend that particular thing that declines earliest, would it just affect that, or maybe it would affect all these other behaviors and phenotypes? So that's what we're interested in this lab. And so I'm going to tell you two brief stories about work that my first two graduate students did to attack the two problems that we think are, I found most interesting when we were starting to work. And that is our work on cognitive decline and also reproductive aging. So first, Mandy Kaufman was my first graduate, one of my first two graduate students, and she worked on cognitive decline. All right, so to get at this question of what behavior declines earliest, we started taking the worms, and instead of looking at how soon they died, we tried to look at what behavior do we care about. All right, so motility is not really a great indicator, because basically the worms will crawl along, and they'll stop moving, and like 2 days later, they'll be dead. Right? So if they live for 18 days, then, like 20 days later, by 20 days they'll be dead. So that's not a great indicator. Chemotaxis gets more interesting. So chemotaxis is the ability, so these worms, they can't see, but they smell. This is how they track down their food. They have really highly developed sense of smell. And so that's been studied for a long time in the worms, so we just picked a chemical that we know that the worms like a lot, and we asked, how long can they do this behavior of going to a spot of that chemical? That's called chemotaxis. And that's what's shown here. And that declines before motility does. So there's a period of time where these worms either can't smell or they can't decide to move, even though they can crawl just fine. All right, so that gets more interesting. So we thought maybe we could build on this ability to chemotax to try to study learning and memory. So what Mandy did, and let me just tell you a little bit about neurobiology work. So they have 959 cells, and 302 of those are neurons. That's all they get, 302 neurons. And about 100 of them are in this thing called the nerve ring. So this is like a very primitive little brain that they have. And a lot of those, so there's nerve ring is right here, and then a lot of these send sensory processes out to the nose, okay? And we know the reason there's writing on those is because each one of these neurons has a name. And I'm going to be talking about the behavior of a particular neuron called the AWC. Has something to do with the winged ciliated neuron. That's why it's called that. And its ability to sense butanone in food. Okay? And butanone is just a chemical, it's a bacterial metabolite. All right. So we thought of the whole idea of Pavlovian training, Pavlov's dogs, right? Could we maybe teach the worms something? And so the way we did that was we said, okay, let's take worms, and they've been happy, well-fed. This is just bacteria that they're eating. And now we starve them for a little while. We just put them in a buffer with no food. And now when we give them food again, we present them with butanone, which is a chemical we know they can smell, but they don't care about too much one way or the other. All right, so we're trying to get them to remember or learn that they like butanone by pairing it with food. And then we do the simple test of chemotaxis. The funny screen, I can't actually see. Okay, where you put them down at this spot, and then wait an hour and see how many of them go to this butanone spot versus this control. This is just ethanol that's been dissolved in, okay? So that's our chemotaxis index. How many worms go to the right spot minus the wrong spot, divided by the total, and we subtract out any worms that are too impaired to move. All right, this is what we found. Okay, so this is our chemotaxis index. And as you can see, the naive or untrained worms don't really care about butanone. They like it a little bit before we start. But once we paired it with food, now they love it. And so this difference is what we're considering the learning index. All right, so this goes way up. The maximum they can get is if 100% of the worms went to that spot. So this is pretty good. If we don't do the starving, they can still learn. They're just not as motivated. And you have to do the pairing in order to get this. So this meets all the standards for classical conditioning. So the very simple assay, we can say that the worms have learned this association between the food and butanone. All right, so how long does it take them to learn something like this? Well, after about 30 minutes, they've pretty much learned all they're going to learn, as you can see here, and training them any longer doesn't get you anything more. All right, so we think that this is kind of maybe something they are useful in their own world. Okay, so we have this learning assay, and we wondered, well, can the worms remember anything? And so the way we tested this was we simply said, okay, so we've done this training stuff. Now if we put them back on food, how long can they remember that association between food and butanone when they don't really have to, because now they're getting the food again? Okay, so we hold it for different amounts of time, and we do this test again. And here you see the results. So they remember something for a little bit less than 2 hours. All right? So we think that's pretty relevant in the worm's world, because maybe in 2 hours their food is gone, right? So there's probably no point in learning something and remembering for too long. So that's what we're considering now short-term associative memory. And there's some really definite, some hard definitions that go along with that, but we're not going to get into that. Okay. All right, so that's short-term memory. And then we wondered, how long can we make the worms remember this thing? All right, so now, that was more like cramming, and this is more like studying all semester. We train them over and over and over again. And then we see right afterwards, did they learn it? They should have. And then we wait until the next day. So 16 hours later, we come back and test them again. All right. So doing this 7x training. So now this is the number of training blocks. You can see like training more obviously doesn't get you anything more. They just, they learn everything they can learn. Now, if we train for 4 training blocks and we come back the next day, they've already forgotten that. But as we get higher and higher up to 7 training blocks, now is as if they've just been trained. So those worms were trained yesterday, they can remember that association today. And so that's what we're using as our standard now. Whenever you see the blue lines, that will be 16-hour memory. And some of you may be wondering what happens if you wait longer. If you wait longer, you train them more, they can start to learn, remember that longer. So it seems to be sort of correlated with how much training they get. So now we're using the 7 training blocks. 16 hours is our standard. All right, so now to be considered real long-term memory, there are some requirements. So that is the requirement for protein translation and for gene transcription. All right, the way we did these experiments was we just simply, during the starvation period between training, we gave them a drug that would interrupt whatever that is. So for example, if we added cyclohexamide, which blocks protein synthesis, now you can see they learn just fine, but they have no long-term memory. They've made no proteins during this time, so this was all just relying on what was there. And the same thing for actinomycin D, which is over here. This blocks gene transcription. So this tells us if the worms can't make any more new mRNA, express any more new genes, it also doesn't have long-term memory. So the reason we did these was not only because we were interested, because this allowed us to say this is just like long-term memory in higher organisms, like in aplesia in flies and in mice and in rats, where this has been well studied. So worms are doing the exact same molecular process as has been studied in those organisms. All right. Now, one of the other requirements for long-term memory in other organisms, including mammals, is the activity of this transcription factor called CREB. So CREB is the cyclic AMP response element binding protein, which simply means when cyclic AMP is activated, there's a whole series of reactions that end up with now another transcription factor here called CREB getting phosphorylated, it becoming active. Okay? So we knew that was important for CREB in these other memory in these other systems. So we asked whether it was in worm. All right. So what I'm showing you here, every time you see CRH1, that's the CREB homolog in worms. So that's the CREB mutant. So this, it means that the gene has been deleted in the worm, and so it's not functional. So a CREB mutant in worms has no problem learning. They can learn just fine, and their short-term memory is actually really fine as well. But what they can't do is they don't have any long-term memory. So we have two different mutants of that in that show. So again, this meets all the requirements in other organisms. And the reason we did all this stuff is because I was really surprised when I started my lab that nobody had tried to understand long-term memory in worms. I thought that was really amazing, and so these are just sort of a checklist of things you have to say, yes, this is exactly like another organism, so we could go on and study something else. All right, so it's just like other systems. And then we wanted to know where's the CREB acting? So we took that mutant that has no CREB anywhere in its body, and we added it just back to the neurons. And the way we can do that is through this trick of expressing it just in a pan-neuronal promoter, and you can see it totally rescues memory. So that again tells us that the CREB is acting in the place where we'd expect it to act in order for us to get memory. All right, so this was all a long-winded way of getting to the point of what I really wanted to get to, but it turned out, since nobody had done it before, we had to figure that out, of asking what are all the cognitive abilities and how do they decline with age? All right, so now instead of showing you out to 3 weeks as I did in those other graphs, you notice this is just the first week of adulthood, so 7 days. So you can see on that day, the worms move just fine. They're all very happy on the plate moving around, and they can chemotax just fine as well. But what they can't do anymore is they're having trouble with the 7x learning. So the more, by 7 days, they really can't do that. They really can't do 1x learning, just that mass training. And finally, their long-term memory is already shot by like day 3 or 4. So this was shocking to us because the worms look fine. They look great. You wouldn't be able to pick out a day 2 or 3 worm from a day 1 worm if you tried. But they already have lost this ability to do this behavior. And so we were actually happy about this. I mean, for us, that's not great, but in the worms, it means that what we can do is start to do genetic experiments because they only reproduce these first couple days as well. And so it's really hard to do genetics on an organism in a post-reproductive period. But this means we can start doing experiments and try to extend this and look for mutants that can do. And also, we can start doing experiments where we add drugs and see if we can push this later and do them relatively quickly. All right, so some of you should be wondering what's going on with the DAF2 mutant that I told you about. All right, so what we did was we took that DAF2 and we tested it for these behaviors I just told you about. All right, so this graph is telling about learning on this axis of short-term memory. All right, and the red, I'm trying to always keep with the red as being the DAF2 and blue being DAF16. So you can see the DAF2 mutants have wonderful short-term memory. We had to extend this out much further than we expected because they don't even lose their short-term memory by hour 6. So this is 3 times what we see in wild type. Okay, so these DAF2 worms, they look beautiful, they have really nice memory. And it's dependent on DAF16 because those guys can't learn and they have no memory at all. Moreover, this long-term memory is totally dependent on DAF16, and we can see in the DAF2 mutant, they really have a nicely extended memory. So out here at 24 hours and at 40 hours, they still haven't forgotten what they got trained to do earlier. So we love these worms. They're just fantastic in a lot of different ways. Now, one thing is they do something slower. So one might be wondering, well, maybe they just, they learn it slower and then they forget it slower. But what we can see here is that they actually learn at the same rate. Now, they don't learn faster, but they learn at the same rate as the wild type worms. So it's not just that they're picking things up slower and then losing it slower. And in fact, with long-term memory, we don't even have to train them as long. Within 5 training sessions, they've already maxed out that 16-hour memory. So they're actually really better at all these activities. All right. So there are different types of longevity mutants. So how many of you have heard of caloric restriction? Anyone? No. And these worms are not, the DAF2 worms are not calorically restricted. They actually eat as much as they want to. But we do have a mutant that mimics caloric restriction. It's called EAT2, and actually, because it doesn't eat very well, it actually can't pump its pharynx, its throat, and it can't chew up bacteria very well, so it's naturally calorically restricted and long-lived. All right, so we wonder what those worms would do in this assay, because we'd like to know if all longevity treatments are the same. But in fact, as you can see, these worms, they learn fine, but they don't have good short-term memory at all. Remember that DAF2 is way out at 6 hours, right? So those are not the same. And in fact, their long-term memory stinks. It's like 60% of wild type. And so that's perhaps the first take-home message. Caloric restriction is not all it's cracked up to be. Yes, exactly. That's how I felt. All right, so then we wondered, okay, so they're not all the same. All right, here's the problem. We got to the point where I'd say, okay, I told you about what's going on the first day of adulthood, but what we really want to know is what happens with age. Okay, and this is the first time the DAF2 worms ever let us down. All right, so what I'm showing you is, on the first day of adulthood, wild-type worms, they have this nice memory, and by day 4 it declines. Oh, sorry, that's learning. And it really declines. The long-term memory, it's shown in here in black, really declines by day 4. But the DAF2 worms do exactly the same thing. And we really thought this would be extended. And instead, it drops, just like wild-type. So that's the only thing that I can find that DAF2 worms don't do better. And on the flip side, with caloric restriction, they start out bad, but they sort of stay at that same level. So they start with this low memory at day 1, but they don't lose much by day 4. So I think the take-home message from this, if I were really to do caloric restriction, I would wait as long as possible, and then I would start it. When you start to feel like you really have to. Because it might help in the end. Okay. All right, so caloric restriction really seems to maintain that long-term memory, at least this low level with age. All right, so we couldn't figure out how this all came together, right? I'm telling you all these different things and probably can't even keep it straight. So what is the underlying principle that that explains all this stuff? And now we come back to Krebs. We looked at a bunch of other genes, but basically everything I'm showing you here says that Krebs levels are what determines that memory. So when we looked at Krebs levels, expression levels in their various mutants, and I should tell you, we have all this microarray data sitting around in my lab from various experiments, so we just started digging around in that. So if you look at Krebs levels in DAF2 mutants, they're higher than wild type on day 1, and they have better memory. DAF16 has lower Krebs and it has worse memory, etc. And EAT2 starts out with lower memory and lower Krebs levels. And with age, wild type and DAF2, the Krebs levels drop, but EAT2 doesn't really drop. So if we start looking at the correlations then, we can say, what are the ratios in any two, like a mutant? Any two mutants, what are the ratios of their Krebs levels and what are the ratios of their memory activities? And we can see there's a pretty nice correlation. And that's what this R-squared value tells us. So this is like relatively correlated. It gets better if we start looking at the actual levels of the protein, the phosphorylated activated Krebs levels in the worms. So that's what's shown here. And that's a really nice correlation. So the best you can get is 1. And so we really think that the Krebs levels are what actually determines the ability for these animals to remember something. So it also tells us that if you have a mutant and you look at the Krebs level, that should predict how well it'll be able to do memory at any time. All right, so what does having more Krebs do? Well, the thing is, it makes everything better also. So here we have wild type. You see, wild type worms lose their long-term memory by 24 hours. But here, you can see that when we have more, so this is now we've made an overexpressor of Krebs just in the neurons. So we put in more and more Krebs, and we can see it boosts it, so by 24 hours we still have them forgotten. And they're also a little bit smarter because they don't require as much training. Here by 4x training, they've already learned everything, or they can remember everything they need to. And it's this is 5x training, just to show that wild type keeps going up and the overexpressions are already there. So basically, the more Krebs you have, the better it is. And so that's certainly true in these worms. The more Krebs any one of them has in their neurons, the better they react. Okay. So I started out this talk by saying that we have this transcription factor called DAF16. It regulates all these genes that help things live long. And so here's another transcription factor, and we wonder what's that thing doing? And so again, we use microarrays. Do you guys know what microarrays are? Microarrays are basically, in my case, so I built a bunch of these when I was a postdoc. They're glass slides, and they simply have spots of DNA for every single gene in worms. So I spotted like these 18,000 genes onto a glass slide, and then I take the RNA out of the worms and I put that onto the slides, and you can say, like, if you compare any two using fluorescence, you can say this gene is higher in this mutant than this one. That's all it is. It tells you levels of every single gene. And that's how we get these stripes, because every single gene is a stripe here. So all we've done here for this experiment is we've taken worms that haven't been trained and compared them against the worms that have been trained. And we just mash them up and get their RNA out and slap it on a microarray, and that's what we get. Okay. The reason this tells us something is because we can do these nice experiments where we set up the experiment with a hypothesis. And this hypothesis is, if we think that Krebs is doing something, we should look for the genes where in a wild type worm the genes can do something, in this case go up, but they can't do it when Krebs is not there. So this is the group of genes that we're interested in. This is called a cluster. And all I'm showing you here is that we get back these same genes when we put that Krebs back into neurons. So we think it's all in neurons. And these pictures are pictures of some of these genes in the worm with GFP, green fluorescence protein, stuck to it. And what we can see from that is that you can see all these nerve rings are really lit up. So that's just telling us that Krebs is controlling a bunch of genes that are expressed in the neurons. This may not be surprising to you, but I should tell you, Krebs is expressed everywhere in the worm. I mean, it's present in the entire thing. So there's no reason that it had to just be in neurons, but because we've done this special training for memory, it seems to really be lighting up genes in the neurons. And this may be very hard for some people to see, but all you should know in this is a picture of sort of our really badly drawn neuron, because we're actually not neurobiologists. We're learning how to draw these things. So this is a cell. This is the nucleus. And here's, for example, a synapse. We have a drawing. And everything you see in red is a gene that Krebs turns on when we do this memory training. And there's a whole bunch of them. There's like about 200 genes altogether. But what's cool about this is about half these genes, we actually knew what they were already. And other people had already said that they have something to do with learning or memory or or neuronal regulation, like sitting at the synapse. And so this verified for us that Krebs was doing something really important. But I have to say, the reason we started these studies is because apparently no one had done too much work looking at what Krebs regulates in any system. And that was shocking to me as well, because people had done one gene at a time. So you could write a paper just about this one gene being regulated by Krebs. So what we decided to do is take this list of genes and say, well, how important is each one? And the reason we did this is because if you remember the DAF16 target genes, they just affected longevity, but in a little manner, right, 5 to 10%. But what we see here is that when we knock out any single one of these genes, we totally lose long-term memory, okay? And I'm just going to click through this because this is basically true for not just synaptogenesis genes, cell adhesion genes. You get the picture, right? That basically you knock out one of these genes and you lose all of your memory. And this is crazy to us, right? Because it suggests that this machinery is really complex, but every single part of it is absolutely necessary. Okay, so more of the same. It's all important. The other thing that these worms do is, so the Krebs is turning on the genes that actually act upstream of it to activate it. So what this means is that the more Krebs you have, the more activity there is, the more it turns on genes, and the more it turns on the things that turn itself on. Right? So it's creating this sort of loop, we think. And that's why, I think, when we do more and more training, we see this steep rise in its ability to remember, because it's basically turning on more things to be active. This also suggests to us that if you don't get activity, say after 4 training sessions, and you stop, it means it hasn't hit a threshold and it's going to forget. And sometimes it's important to forget things too. So it has a built-in mechanism to make sure something is important enough to remember. All right? And so again, if we knock out any of these genes up here, we also lose each long-term memory. Oh, sorry, and this is what I was trying to explain. With more and more training, we see there's more and more Krebs, there's more and more memory, and we see more of the reporter for this. Okay. I'm not going to dwell on this too long. This is just a list of brand new genes. So I told you about all the genes that people already knew about. But when we do microarray studies, we also learn about a lot of new genes that no one had discovered before or no one had studied. And the reason we're interested in this is because these genes are highly upregulated when the worms get trained. They're Krebs targets, and there's mouse and fly orthologs, which means that there's a good chance that it will have something to do with memory in a higher organism. And so when we knock some of those genes out, what you see these black bars, again, we lose long-term memory. So now this is a way for us to discover new components of memory that haven't been studied in any other organism. And so we hope that basically this will be a new way to study memory in higher organisms. All right, so I told you a lot about where Krebs is and that it's expressed in the neurons and you get this memory back. But it occurred to us that we didn't know exactly which neurons this happens in. And so we did this experiment. Basically, these are the untrained worms, and then these worms, and what they have in them is this, this is basically the DNA element that the Krebs transcript factor sticks to. So when it sticks to that, it turns on this green fluorescent protein. So when you do a lot of this training that I told you about, and Krebs gets really activated, what we see is it lights up a very specific neuron. So these aren't just a pair of cells. This cells we know has something to do with starvation. We feed and starve the worms a lot. We don't think this one's important. But this one no one had ever seen before, and it's called AIM. And that shouldn't mean anything to you, and it didn't mean anything to me either when I got it. But we think we were surprised at how specific that activity was. And so what we did was we thought, okay, well, if it's a really important neuron, like if it's really the equivalent of the human hippocampus, then it shouldn't matter what kind of learning we're doing. So we decided to switch it up. We changed it, did that even in food training, and then we tried it with all different conditions. Anything that was basically an associative memory, where it learned two things, to associate two things, lit up the worms in that same neuron. So this leads us to believe that this is basically where the memory happens in worms. They do it in one neuron. They can't do calculus, but they can actually, it seems like they can make some sort of, form some sort of memory and keep it there and store it in that neuron. And when we knock out that neuron, that's what these two mutants are. Again, we lose that long-term memory. All right. So what I told you, I'm just going to wrap up this story about cognitive, is that we have this transcription factor called Krebs that gets activated, and it turns on a whole slew of new and known memory genes. And so we're hoping that that list will help us learn something about memory in all organisms. And then it does it through this cool activation through like multiple training sessions, and we think it's because of this feed-forward mechanism. We know a little bit about what neurons it's involved in, and this is kind of cool because this is the sensory neuron that senses the food in the butanone, and it somehow transmits the information a couple of neurons down. And so we're trying to figure out what that communication is. And finally, we know that the Krebs levels really are a direct mechanism for turning on that memory. And that's exciting because that's a transcription factor that exists in all of us. All right. Now I'm going to tell you about a little bit of work that my graduate student, Shivani Guo, did. And she was also, I had these fantastic two graduate students. They started with me as rotation students in 2005, and they just, man, did a ton of work, as you can see. All right, so the reason we're really interested in reproductive aging is because I feel like it's one of the understudied fields in aging. We focus a lot on very, very late aging, but human reproductive capacity declines extremely early, right? So it's one of the first, it's probably the first aging signs. So for example, in the mid-30s, women experience a greater incidence of infertility, miscarriage, and chromosomal abnormalities. And this in humans is thought to be due to declining oocyte quality. So we thought this is a kind of interesting problem. If we, instead of looking at just at lifespan, now we're going to look at something called reproductive span. So how long can an organism reproduce? And women have this really long post-reproductive lifespan, which has been attributed to many things, including something called the grandmother hypothesis. But we realized that worms actually have almost the same kind of profile, which seems weird, and this was always chalked up to something in the lab. And so we wondered if there was some sort of relation between them. Could they be caused by the same thing? Okay, you're not going to have to know too much about this, but I am going to tell you a little simple thing about the worm reproductive system here. Because really, that's all worms, they eat and they reproduce. So that's why this is like their entire body. Part of it's intestine and the rest of it's making eggs. Okay, so what they do is they have this germline that consists of, these are mitotically proliferating nuclei. These are just cells that are replicating. And then they're going to become packaged into oocytes here. And then those oocytes will eventually squeeze through this spermatheca, and this is where the sperm sit. So these are hermaphrodites. They make both sperm and oocytes. But we can cause them to not, we can knock out these sperm and they can just be mated with males. And so they can essentially be males and females. Right, so they squeeze through here and there, they create these fertilized eggs. Now they have a lot more germline stem cells than they'll ever need. And so we don't think that's limiting anything. And they make about somewhere between several hundred, like 300 to 1,000 eggs from any one mother. So that's what all you need to know about worm reproduction to understand this topic. Okay, so when I started this work, I was talking to people who work on worms, and they were like, oh, if you just wait longer and longer, you can just get them to have a longer reproductive span. So while we did this experiment, but actually Carrie Kornfeld's lab did a really nice version of the same experiment. So I'm going to show you his data. And that is, they just took worms, and here's wild type worms, and they took 2 mutants that don't make sperm. So they absolutely have to mate with a male in order to get fertilized. And what they did was they mated them later and later and later. And instead of seeing a big shift, like if that holding on to them actually helped, then you should have seen a peak just going to the right more and more. Instead, what you see is that they just don't fertilize after a while. So holding on to them longer doesn't help. Does that make sense? All right, so has nothing to do with the number of the oocytes you use. They call it usage independent. Really, what it means is, like humans, it's a use it or lose it mechanism. Like, I can't have a baby at 80, right? Because I'll be put out here. So that's what this has to do with. These oocytes are not getting in, not staying good. Now, we knew that we could actually extend this reproductive span because the DAF2 mutants that I told you about a lot, every once in a while when we're doing a lifespan assay, there'll be an egg that pops up on the plate. And that doesn't seem possible, but except that they can actually reproduce pretty long. They can reproduce about double the time of wild-type animals. And those calorically restricted animals can also do this. So there's some connection between long-lived animals and their ability to reproduce later and later. So we wondered if there's other regulators that we didn't know that are out there. And so what we wanted to do is relieve this restriction of saying they have to be longer. We just wanted to ask, can anything reproduce longer? And so to do this, what we actually did was we went to the mouse literature. And someone else, this Hamatani et al. Paper, they had a nice piece of work where they had taken oocytes out of young worms, I can't always say worms, young mice and old mice, and they compared the gene expression. So basically looked at what changed with age, okay? And they mostly paid attention to the genes that go down with age, but we noticed in their list, there's also a set of genes that go up with age. So we thought these things could be bad. These things could be accumulating in the old oocytes. And one thing we noticed right away was there were a lot of genes involved in TGF-beta signaling, and actually there's more than they even put on their list. And so what we did was we said, well, is this going on in worms as well? And to do that, we just got mutants in that same pathway and started studying them. And lo and behold, that actually turned out to be exactly right because we took all these mutants. So now I'm showing a different pathway, much like the insulin pathway. We have a receptor and a bunch of signaling that ends up affecting transcription. But in this case, and I apologize for the nomenclature. The reason they're called SMAs is because the animals are small, and they're the SMAD pathway, so that's what some of this stuff is. But all you need to know is there's a ligand called double-1, which we're actually not going to talk about much. And there are mutants here, and every single mutant in this pathway extends reproductive span really nicely. Okay, so I was really excited about this because it suggested something straight from a mouse study that we asked about in worms. It did exactly what we wondered it could do. And this just shows you don't have to, the sperm have nothing to do with it because you can take wild-type males, so the normal sperm, and mate them with these mutants, and it extends reproductive span as well. Okay. And I want you to remember this point. These worms are happily reproducing, and then all of a sudden they die. And they die because the eggs hatch inside the mother. It's called matricide, and it's gross. But I'm going to come back to this point. All right, so not all is well with these mutants. All right, so we wanted, okay, what are we looking at? They can extend reproductive span, but does it have anything to do with what we originally asked, oocyte quality? So we did some very simple tests. The first one is if you just take worms on the first day of adulthood and the 4th day of adulthood, and you bleach them to get their eggs out. So bleach is literally it's exactly what it sounds like. You wash them with bleach. That dissolves away everything else except for these chitin-protected eggs. You can see that on day 1, every egg you get out of that treatment will hatch. But if you wait until look at day 4 mothers, those eggs have less of a chance of hatching. Okay, so already we're seeing just in a gross manner some sort of decline. But the real question is, can they get fertilized? Because that's the question for humans. Okay, and a fertilized embryo obviously ends up looking like an egg. Worms will also lay these unfertilized oocytes, these sort of mushy little things on a plate, and we can start to count those. So we see in wild-type worms, you get a bump, you start seeing some of those before they completely peter out. But in DAF2 worms, they only lay fertilized eggs. They never start to lay unfertilized oocytes. We really looked hard for them and we never saw those. And the same thing for those TGF-beta worms. We see, like, I think there's one here. Okay, so they just always fertilize every single egg, that oocyte that they have. So that already, their fertility seems to be better. And then if we look at just the shape, I told you these animals are transparent, so we can just take a microscope and start looking at the different parts, and we see those oocytes are really nicely shaped in the first day of adulthood, but they start to look pretty bad by day 8. So they've got small oocytes and these gaps in between them, and then sometimes they like squeeze through the spermatheca even though they haven't been fertilized, and form this giant oocyte cluster. So my students counted all of these things and scored them, and you can do something called a non-parametric analysis to try to decide whether things actually look better or worse. And the yellow means it has a severe defect. So you can see that animals that are reproductive look fine for all these capacities, but if they're post-reproductive, if they've already stopped laying eggs and oocytes, they start to accumulate some severe phenotypes, especially this clustering phenotype. So wild-type worms with age accumulate these defects, but the DAF2 worms and the small tooth, so the insulin receptor and the TGF-beta signaling worms, never look really that bad. They keep their oocytes looking great the entire time. And that's scored here. You can see a lot more black bars because they look more normal with age on this day, 8th day of adulthood, which is really remarkable. It's pretty late in their life. We can also count their chromosomes, which is really what counts, right? If we're thinking about humans and chromosomal defects, aberrations like Down syndrome. And so worms normally would have 6 chromosomes. And if we count how many of them have the wrong number of chromosomes, we can see that that's, or here, the right number of chromosomes, here that declines with age, and that never goes down in the DAF2 worms. And that, of course, has some functional effects, right? If you lose an autosome, one of your normal chromosomes, that egg may never hatch, will never hatch, right, because it's in bad shape. That doesn't happen to the SMA2 and DAF2 worms. And a kind of cute thing in worms is that they lose their X chromosome, they're fine. They'll just be a male. And so we noticed that a long time ago that we get more, no one had to quantify this, but if you just look at the progeny, there's more and more males with age. And we think there's actually good evolutionary reason for that, because they want to have some sort of outcrossing as they get older and have more problems. But the DAF2 and SMA2 worms never do that as well. So we think that they're actually not losing their chromosomes. They're actually paired correctly and passing them on correctly their whole lives. Okay, so this is for the same. The germline also looks good. I'm not going to go through that. So basic message is DAF2 and SMA2 worms look better. And so the bottom line is that in these animals, we see that with age, there's more and more mitotic germline quality decline, and that oocyte quality is really getting bad with age. And we think that that's what leads to reproductive aging. And that's what we see reversed in the animals that can extend their reproduction. Okay, so you're sensing a theme here because we're going to go back to microarrays, because what we really want to see is what are all the genes that are different in these mutants. And so Cheating did this really heroic experiment where she took worms and she chopped them up with razor blades and filtered out their oocytes. So that's what she got here. And did these very nice microarrays to look at what changes with age and what is different in these worms that have the better oocytes. What's different in the actual oocytes? Okay, I don't expect you to read this. This is a daunting slide, but all you need to see here is these are up. So they're genes that are way up in the good SMA2 oocytes. Okay, and the reason I show this slide here is because you see a lot of these names look similar. The reason these names look similar is because these are exact same genes, the homologs in the worms and in humans. So this is the mouse study I referred to before, the oocyte aging study. There's one human oocyte aging paper that we could come up with. And these are genes that are basically high in the TGF-beta oocytes, and they decline with age, and they decline with age not only in worms, but also in the mice and in humans. Okay? And the kinds of genes that are coming out of this are interesting because these all have to do with chromosomes, keeping the chromosomes together, keeping the DNA repaired, everything that has to do with chromosomes and DNA repair, which I didn't talk about for the somatic aging, if you'll recall. Okay? And everything in a red box means this is actually the same gene. So this was way beyond our wildest dreams. When we started this project, what we thought was we'd find some things that are kind of similar and might be nice, but these are exact same genes in all these systems. So I'm really mystified by why worms do this and have exact same genes changes in humans and in mice, but it does suggest that we can learn a lot from them. All right. So one of the quick things, just to test to see if they're actually important, what we did, we took this list, we tested them for how many of them affect things like embryonic lethality, so hatching, And then he took some of our favorites, like this condensin that was this is the same gene in humans and mice, and we tested them for how many of them laid unhatched embryos instead of the hatched embryos that we like. And you can see that's terrible, right? Compared to wild type or the control, these animals are really screwed up. And they're really screwed up in a lot of ways. So just losing that one gene that holds the chromosomes together can really mess up their germlines. That's in some ways not that interesting because we kind of knew it would anyway. What's interesting to me are these kinds of genes where you see out at day 10 and 9, these look much worse than the wild type. And so what that means is that, and they even have unfertilized oocytes, within this set of genes are the genes that actually are age-related. So they're more to do with age-related decline, and so I want to follow up on those a lot. And some of them, for example, are specific for repairing, this is gamma radiation, so DNA repair gene. All right, so what I've told you about is that you have these decline the germline and the oocytes, and it's through these, the regulation of things that are important for keeping the chromosome and chromatin and DNA healthy. Okay? I skipped over a lot of this, but there's interesting signaling that goes on. So the animals seem to regulate the rate of this decline through the rate of signaling by TGF-beta and by the insulin-like peptides. And the reason this is interesting is because basically they're sensing their environment and taking in information about food levels and their neighbors and what the environment is really like to be able to regulate the rate of that aging. And that's what I find interesting because they're able to modulate that. It's not that they're just like marching ahead always. They really do adapt to their environment. And the reason that gets more interesting is because Cynthia, my postdoctoral advisor, Cynthia Kenyon, she had previously discovered that the rate of mitotic proliferation actually affects somatic aging. So she had done this crazy experiment where they knocked out the germlines of the worms and then saw that they lived longer. And at the time it didn't make a lot of sense, but now I think we have a nice way of thinking of that, is that if the worms sense that there's not enough food, what they might want to do is slow down their reproduction so that they can lay eggs into someplace where there is more food. So they can go someplace where there's more food. But it's not going to do them a whole lot of good if their bodies are already falling apart. And so what I would like to suggest is that they modulate not only the rate of reproduction, but then they have to tell the bodies to also extend lifespan in order to have this nice cushion of a post-reproductive lifespan. And the reason they need that is because those mutants that I told you about earlier that die in the middle, it's because their bodies are falling apart. So these worms, their reproductive system is in great shape. Their oocytes are great, they're laying eggs, but their muscles are too weak to actually lay the eggs. And so it's important for organisms, I think, more than just probably worms, to be able to tell their bodies that they need to stay in great shape because the process of childbirth is not that easy. And so if it's falling apart too early, they're not going to be successful their entire reproductive span. And just the last thing I want to hit is that I told you a lot about the DAF2 worms. So we actually looked at their oocytes as well. And as I mentioned several times, in the oocytes, like this is the set of genes that they really care about: DNA repair, cell cycle, chromosome, and chromatin maintenance. Whereas if in the somatic genes, those are an entirely different set. There's just really no overlap. So what we're seeing is a bunch of genes that are important for proliferating cells, cells that can replace themselves, such as our stem cells, and these genes that are important for keeping the cells that we're stuck with our whole lives. You know, so our brains and our muscles largely cannot be replaced, and so we have to care about these genes for those tissues. But other tissues, such as our skin, can be replaced, and so those cells really need to keep their DNA repair and chromosomes working well. All right, so there's different programs for aging of mitotic and post-mitotic cells. All right, so just to wrap up, what I told you about today is that we think we found basically a little hippocampus in the limb, and where they keep memory, and we found what we think is the earliest decline that we can track in these very simple organisms. And we have something that we'd like to think of more about how similar are the declines in reproductive aging in both humans and worms. And finally, this idea that there's two different types of aging. The types of aging that goes like cells that can repair, turn over, and cells that can't. And those require very different maintenance systems. All right, so these are the people who did the work. Really the stars. That's Xi Jing dressed as a worm. And Andy. Actually, this was a GFP worm, and just the lights were not off. There's still little lights in it. So that's Shooting Low. She did all the work on the reproductive aging with some assistance from Gunnar and Wendy and Jasmine. And Mandy Kaplan really developed all the work in the learning and memory with the aid of some excellent other people in the lab. And we get strains from a lot of people. The worm community is very generous, and we send strains around everywhere. And we get nice funding from different people. So I appreciate you listening, and I'd be very happy to take any of your questions.
You talked about knocking genes down, or what do you use to turn the genes on or off, or kill a gene, or put a gene in?
That's a great question. Okay, so one of the great things about worms is that we can do something called RNA interference. So basically what we do is we feed them bacteria, and the bacteria actually have a circular piece of DNA with our gene of interest, and it's flanked by this promoter, T7 promoter, and what it does is it makes double-stranded RNA. Okay, the worms eat the bacteria with this double-stranded RNA in it, and through a process that's really being carefully studied now but not totally understood, that message will go in and turn on a system called RNAi and basically degrade the messenger RNA that's in the worm's body. So that's a really fast way for us to do experiments. And it's something that only worms can do it by feeding. Other organisms, you can inject the double-stranded RNA and it does the same thing. And plants do that as well. And so we use that as a way to turn off genes very quickly. And that really started, I was very lucky because I was one of the first people to get to use that when I started doing the lifespan studies. The other way, the more traditional way, is to just make mutations. You basically use EMS as a mutagen, so it changes DNA and you look for the mutation in that gene. And we also do something, if we want to add something back, like I told you about the Krebs studies, we make the DNA for that gene that we want, and we actually take a needle and inject it into the gonad of the worm, and it takes it up and makes more of it. So we have all kinds of tricks for dealing with that.
Will this apply to humans eventually?
Which part of it? Look, I think the best thing for us to do is not to look necessarily, is to know what genes we're interested in and to understand how those work in humans. So we can start working with human cell lines, for example. But what we're really interested in is changing all these into really high throughput systems. So I have a whole slew of undergraduates working on crazy things like building little robots to do assays. Because what we want to do is be able to test chemicals, okay? Because what we would like to do is screen through like 1,000, we have a set of like 25,000 chemicals, and ask which ones can extend lifespan, which can extend memory, and which ones can extend reproductive span. And hopefully not kill the other ones when they do that thing. So that's the kind of thing, I think that's more useful for finding what we can do in other organisms.
How sensitive are your measurements to temperature variations?
That's a great question. So we do everything in very controlled environments, but the worms themselves are very sensitive to temperature. For example, that DAF2 mutant, the way it was discovered was through a mutagenic screen that looked for animals that go into a special state called Dower at 25 degrees Celsius. So we work at about 20 degrees for most of our experiments, and they can be raised at 15. But we try to keep an eye on that because they have entire sets of neurons that are dedicated just to sensing temperature. And I think if we don't take care of that, then it can really screw up our experiments. Yes?
Can you swap in human or mouse genes for the worm genes and see if they behave the same way? And some of these targets have drugs that have been made for years, but maybe not done it.
Yeah, so we're actually interested in the second half of the drugs. Yeah, absolutely. Especially because the NIH has a whole set of drugs, chemicals that have already gone through different phase trials, right? So you can actually test them. But knocking in genes, is that what you asked about? Knocking in genes? See, my memory is... Yeah, swapping them. Oh, for example, the human insulin receptor has been put into worms, and it actually activates that DAF2 insulin receptor. I mean, sorry, the human insulin was put in. So we know that those can actually put in. So we do it all the time. And in fact, actually in work I didn't have time to tell you about, we have done experiments with human tau. So tau is one of the proteins that's involved in Alzheimer's disease. And so we have, and I'll show you something called 17 frontotemporal dementia, which is actually even more tragic. But so the human normal tau we put into worms, and we have the two different mutants of tau. And the two mutants of tau have terrible memory. And so while the control is normal. It's fine, it's okay memory. And those 2 mutant forms can be partially rescued by the DAF2 mutation and also by treatment with resveratrol. So we're trying to understand now, why does it fix it? Does it fix it by changing the aggregation, which seems to be part of the story, or is it there some other phenotype? But that's exactly the kind of thing that we're doing with human genes. Yes?
Your worms have been evolving for a rather long period of time. In a Darwinian sense, evolution for optimal performance. They have a very short lifespan. I guess what I'm trying to ask is, if you make them live longer, are there downsides to that? The worms that live longer have problems, or in other words, why? To be facetious, why did Darwin make them live longer? Must be some problem.
So the underlying, so there's a principle called antagonistic pleiotropy. So basically, it's something that's good later, probably bad earlier, and vice versa. And in fact, actually, the reason DAF2 are mutants and not the wild type is because they will always lose out in the natural environment, because they take a little bit longer to go from an egg to an adult, and then they have about the same number of progeny, slightly fewer, so that's a disadvantage as well, and they have them over a longer period of time. So even though I'm interested in extending reproductive span, for them, that's a disaster, right? Because they will already be on, like, so when wild type, if you just go to start 2 eggs, start them a race, wild type, DAF2. Wild type already gets to be a reproductive adult here. It takes a half a day longer. It has way more by the second day. This guy's still behind. So by the next generation, it's had 300 already, right? So it's going to keep expanding before this one does. So that's why, because it always takes the DAF2 a little bit longer to do everything. And the long life doesn't help it. All that matters is reproduction. How many of its genomes it gets out into the next generation. So if it has more, if wild type has more, and it has them earlier, it's going to win the race. Now, the reason there's this change, so the environment, right, they have to be able to react to it. So in times of like high heat, low food, under those conditions, the DAF2 worms might do better, and because they can actually like survive that better. But the wild type worms are actually in this middle place where they can actually go up and down because they need to be able to. I mean, the evolution did happen. They have like all these worms that from the wild type that can get there first before DAF2. The DAF2 just loses out. But for, we're anthropomorphizing this, right? We're saying that like living longer is better, reproducing longer is better. It's not better for them. Yes.
Has that been shown that under conditions of low food or high heat that DAF2 actually could evolve? DAF2 could evolve?
So David Gems did a series of experiments with that, and Gordon Lithgow did as well, but only for like 10 generations, just to establish the principle. So that's as far as I haven't been interested in following up on that, but it has been addressed.
How much is known about the similarity of the memory you talked about in worms and memory in higher level organisms? Those mechanisms similar?
They're very similar. And in fact, what I wish I knew more of in other organisms was downstream of Krebs. Because everything I can find that's downstream of Krebs in mice is in our list. Our list is much bigger though. So they all require Krebs, and for example, if you do this kind of training in mice, they can remember it for like 24 hours, so not a whole lot better. So it really is very similar. Now, if you're talking about decades long, I think that, I don't know yet. Like, we have to figure out exactly, but everything that's known in mice so far, for example, we found in our list. So I think it's a pretty good comparison.
Okay, well, let's give her a